We now turn our attention to the way that cells divide, how they reproduce. And a majority of the cells will reproduce through a process known as mitosis, which is going to be the subject of this particular lecture. There's another form known as meiosis, which we'll talk about in a future lecture. Before we can talk about cellular division, I want to unpack a little bit about the nature of the genome within a cell, because if you can imagine a cell dividing, it has to create basically double of all of its parts, and one very substantial part is the genome. Again, genome is like a huge, vast library full of instruction manuals that the cell can reference to build anything that it needs to build inside the cell. And so there's quite a bit of energy and effort that the cell will take to copy and make a duplication of that entire genome so that the cell is then ready to divide. So what is this genome? How is it composed? We already talked earlier that it's made out of DNA. DNA, again, its features are that it's double-stranded, and it has bases that are in the middle. The bases in the middle undergo what's known as complementary base pairing. And that means that T's always bind with A's, C's always bind with G's, and vice versa. This system ensures that the DNA which is copied is going to be accurate. And it's not always accurate, and that causes problems, which we'll mention later in the lecture. But in either case, we have two strands, complementary base pairs in the middle. It's the sequence of these bases that serve almost like letters that provide the information. So in the same way, we have letters that we arrange to make words. Those words can be used to describe something or provide information for something. Similarly, the sequence of bases can be arranged in a nearly infinite array of combinations to make all sorts of different types of protein. Ultimately, that is what the thing is that is built from DNA. It is this very versatile molecule known as protein. Okay. So that's what DNA looks like. DNA, again, stands for deoxyribonucleic acid. Deoxy refers to the fact that the sugar that is found in this ladder is the sugar deoxyribose. Okay, one other thing I'm going to have you note from this picture, you can see that the strands, each strand of DNA is actually arranged in opposite directions. So like on the left hand side, we can see that the oxygen atom in this sugar molecule is facing this way, but the oxygen atom in the sugar molecule is facing on the bottom on the other side. This is known as anti-parallel when the strands run in these different directions. And that feature of it being anti-parallel is going to pose some interesting dilemmas regarding how this whole molecule is replicated, but we'll talk about that in a minute. Okay, on DNA, there will be genes, and you can think of a gene as just basically being a region of DNA that codes for something. Oftentimes, it's just a particular protein of some kind. So if there's some area, like let's say the area between here and here, where there's a certain sequence of bases that codes for a particular protein, then that would be the protein's gene. Now, not all regions of DNA build stuff. Some regions of DNA are kind of considered spacers, or they serve alternative functions. So not every last base on a strand of DNA is part of a gene, but the regions that do code for things are called genes. Okay, when we consider things that are built, it gets into this duality here, this dual relationship between what is known as phenotype and a genotype. And the way that I describe this is in the same way that a house would have a blueprint, and before the house is even built, that blueprint exists. And then it's the blueprint that determines how the materials have to be put together in order to make the house. That's a little bit about that analogy gets to the heart of the difference here between these two words. The genotype would be like the blueprint. So this is the actual instruction manual, the letters or the diagrams on the blueprint, so to speak. The phenotype then is what's actually built. It's the finished product. It's the thing that we see. Okay. Genotype will be a combination of these letters, or it's the information stored on the gene. The phenotype is the actual thing that you see. It's the trait that you possess. Okay, chromosomes are arranged like this. We've already seen a picture of something like this, but just for reference sake, the DNA is going to wrap around these protein complexes that will form what's known as a bead on a string model here. Those beads all get clumped together to form almost like a strand of these beads. That strand is then folded in and on of itself to almost form a little bit more of like a cord type, stronger arrangement. And then that is in turn also coiled, and then that's what packs into the chromosome. So a lot of coiling that's going on here in the chromosome. A very interesting feature of chromosomes is that they can condense to form discrete little units, as we see here, but other times they can decondense and somewhat unravel a bit, become more loose, and that would allow greater access to the genes that are found along that really long molecule of DNA that's packed and coiled and coiled and coiled. All right, the genome looks something like this. So here is a pretty cool little picture. We're seeing a fluorescently labeled nucleus here, and in this nucleus you can see what the DNA would look like if it was all decondensed and loose. And then just prior to division, the DNA packs into these very nice discrete chromosomes, which are visible. This makes it much easier to make sure that the DNA gets to where it needs to go. Again, you're sorting this entire library, copying it, and then trying to somehow package it into 2 new cells. And that's a lot of material to move around, and to move around efficiently. If just one of these chromosomes didn't make it into the right cell, that would be pretty much the end of the cell. So huge consequence for not having all the chromosomes duplicated and moved to where they need to go. Condensing them into these discrete packets helps the cell achieve the proper transportation of these packets. Okay, so there are 46 chromosomes in most of your cells, but those 46 chromosomes are arranged into 23 pairs of what are known as homologous chromosomes. So a homologous chromosome basically refers to the fact that you have one set of chromosomes for your mom, one set of chromosomes for your dad. Chromosomes 1 through 23 that you got from your mom have homologs. They have partners in the 23 chromosomes that you inherited from your other parent. So the homologous chromosomes will be the chromosomes that are the same size and bear the same traits on them. So for example, on your mom may have given you this particular chromosome and your dad given you this particular chromosome, but they are the same length and they have the same traits on them. Like maybe this one bears the information that colors hair blonde, and this one bears information that colors hair brown. It's not the same phenotype, but it is the same trait, hair color, okay, at the same location. So those would be homologous chromosomes when you have the matching chromosome from one parent to the other parent's sets of genes that they donated. Again, they'll be the same size, they'll have the same traits on them, even if the actual phenotype of the traits is different. Okay, an organism that has 2 sets of chromosomes is considered diploid. And then any cell that has only half that number, just 1 set of chromosomes, is considered haploid. And in a human, basically the only haploid cells that exist are the gametes. And these examples of these would be the sperm cells and egg cells, the reproductive cells. Now this is important because if you consider like a sperm and an egg, if there weren't haploid cells and the sperm and the egg cells had the exact same number of chromosomes as a skin cell, for example, so they both had 46, if these two cells combined together and formed a fertilized egg or a zygote, then that zygote would have double the number that it should. It would have 92 chromosomes, and then on the next time it would be even double that and double that and double that. So there always has to be a reset, and the gametes are haploid, and the way that they are produced is that reset. Diploid cells reproduce to form new diploid cells through the process known as mitosis, which is what we're going to talk more about in this lecture. And then diploid cells create haploid cells in a process known as meiosis, which we'll talk about next time. All right, a little bit about what the cell does before and during and after it's dividing. You can break up the cell cycle into two phases. One is interphase and the other is mphase. Interphase is the longest phase. This is what cells spend most of their time doing. And then just as they're actually undergoing the physical division process, that would be part of what's known as the mphase of a cell's life. So the interphase, we can break that up into 4 different phases. The first is the G0 phase. Oftentimes in a diagram, people will put that phase off to the side here. And this is an interesting phase. This is just basically, the way that I like to think of this phase is just its day-to-day life. It's a cell going about its business. And how long a cell will spend in the G0 phase it varies greatly. For example, some neurons in your body, they don't even really divide ever. So they spend years and years and years just going about doing what they're doing, sending electrochemical gradients and such. So that's the G0 phase. Now, if a cell is then decided it's going to divide, then there's some preparation that must be carried out. The first is the G1 phase, so that would be the phase that we're seeing here in this cell cycle. And in the G1 phase, basically what's happening is that building materials are accumulating. Accumulating, I'll just abbreviate it here. And one of the principal building materials required here is ATP. To copy an entire genome and all of the organelles and all of the other stuff in the cell, that requires a heinous amount of energy. And there needs to be that energy available for that process to occur. So this is one of the first steps here, is just to acquire the ATP required. And the cell also during this time will be acquiring things like amino acids and carbohydrates, all the precursor molecules that it requires to build stuff. As soon as that preliminary gathering of building materials is complete, then the cell will start to photocopy its entire genome. And this is going to occur in what's known as the S phase. So in the S phase, you can say that DNA is replicated. Or, as they put here, DNA is synthesized. New DNA is made. Now, this is a very extraordinary process. Just consider this for a minute. How long would it take you to copy a genome if you did it one letter at a time? It's kind of an interesting question here. Okay, so you're copying it one letter at a time, assuming you slept 6 hours a day. Well, let's imagine that, well, we don't have to imagine. There's about 3 billion base pairs in your genome. And so if we were able to copy one base pair for every 1 second, so you've got, 3 billion base pairs long, and you're just going through, and each one that you see there's an A, you write down a T, and every place where you see there's a G, you write down a C. You've got a template strand, one single strand of DNA, and you're copying letter per letter per letter. That would take you 127 years to copy that genome, assuming you slept 6 hours a day and didn't do anything else. You didn't eat, you didn't go to the bathroom, you just copied those letters one after the next. 127 years. How long does it take the cell? About 8 hours. So pretty fascinating feat there. Now the cell gets to cheat a little bit because it's got these special photocopiers, and it also has many of them. So it'd be, analogous to you and all of your friends trying to copy down the genome. But the cell can do it in 8 hours, and this is a pretty involved process. So basically what happens is the first thing that needs to happen, I guess, is that DNA needs to unwind. And that is done by this enzyme known as helicase. And from there, we have two strands that are now separated out. There'll be some single-stranded binding proteins here that try and keep those open so they don't come back together, and so this strand doesn't come back and get attracted to the other one again, so they don't close up. But in either case, it's like a zipper. It opens up the two strands, and then once they're exposed, the photocopier can come in, which is DNA polymerase. And as the name suggests, polymerase, DNA polymer, ACE, means it's an enzyme that builds the DNA polymer. So it's an enzyme that creates DNA. So what it does is, as the polymerase is riding along here, what's known as the template strand, that would be the one that's exposed, anytime it sees a base of one kind, it adds its complementary base. And so as it just goes one at a time, and it just goes and adds all the complementary bases to the original strand. How it is that it sorts out all those bases and gets them all into the right slot at the right time, that'll be a mystery I'll never be able to understand, but that is what it is able to do. Now, one of the caveats here with DNA polymerase is that it can only synthesize DNA in one direction. But remember, we said that this strand here runs in one direction, and this strand here runs in a different direction. So the polymerase has to work in two different directions because of that, to accommodate that anti-polarity. So DNA polymerase on this strand is going straight up this way, but on this other strand it's actually going backwards. So it's a little bit tricky. It forms what's known as the leading strand and the lagging strand. So in the leading strand, basically DNA polymerase just binds, and it just cruises, keeps adding new DNA. As the helicase unwinds, it just keeps following along it and adding a new strand to the original template strand. On the lagging strand, however, the polymerase has to come up, bind, and then synthesize, and then as the helicase unwinds it even more, it jumps up, binds, and then synthesizes, so it's synthesizing in these short fragments, and then they eventually get stitched together. Now, this is a gross oversimplification of the DNA replication process. In reality, there's things like primase, which are adding an RNA primer, and there's exonucleases, there's ligase, there's lots of different stuff involved here. But for our purposes, this will get us about as far as we need here. We got DNA unwinding, and then the big photocopier here, DNA polymerase, is what's making the DNA, and it's making it by reading a template strand and adding complementary base pairs to that template strand. There's a great video that you can watch, which I believe is just called DNA synthesis. So if you search for it online, I'm sure you'll find it just as called DNA synthesis. Great little review video there, but I'll let you watch it on your own time if you like. Okay, so during the S phase, then, basically how it works is one chromosome will duplicate into two what are known as sister chromatids. So once this chromosome now has a copy that's attached to it, now these are considered to be sister chromatids. And these are identical. They should be identical photocopies. These are not the same as homologous chromosomes. Homologous chromosomes are individual separate chromosomes that, though they are similar because they are the same shape and size and have some of the same traits, they are not identical. They do carry different information on them. Sister chromatids are exact photocopies of the chromosome, and so the one chromosome photocopies itself, and now there's two, and those are called two sister chromatids. And those will be in place. They'll be formed after the S phase. So you could think before the S phase, there's 46 chromosomes, and after the S phase, there is 92 sister chromatids. That's kind of a good way of looking at it. All right, now once the DNA has all been copied, other stuff needs to be duplicated. So the G2 phase, the big thing that's happening here is that the organelles are duplicating as well. So we got two sets of genomes, we got two sets of organelles, and once that's accomplished, then we are ready for the M phase, and the M phase is where the cell is actually going to go through the specific events that cause its division. In M phase, there can either be mitosis or meiosis, and today we're looking at mitosis. We'll take a look at meiosis, which is a bit more complicated later. Here are the phases of mitosis shown. There's prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. Some of the big events that are happening in these different phases. In prophase, we have the nuclear envelope disappears. Another thing that happens in prophase is that the chromosomes condense. So while earlier I'd said that after the S phase you have one chromosome and then now there's a sister chromatid, even though this is true, it doesn't look like this until later on. It looks something quite different when it's before prophase. So in prophase, you're actually getting all of that loose DNA packed and tightly coiled and arranged so that's forming discrete chromosomes. And so that's why we're seeing these here starting to form in the nucleus. Okay, so those are the two events here. Nuclear envelope disappears, chromosomes condense, prophase. In prometaphase, we have a couple of exciting events. One, the kinetochore is going to form. And what that does, it attaches to the mitotic spindle apparatus. All right, so we're seeing in this picture there's these big poles coming out, and they emerge from an organelle known as the centrosome. A centrosome is basically a pair of centrioles. So we learned about the centrioles already. Didn't quite know about the centrosome, but that is going to be what two centrioles form. They extend out these poles. A little perplexing though, because this also happens in plant cells, which don't have these centrosomes. Okay, then those spindles, those long tubes, they attach to this tiny little area on the chromosome, which is known as a kinetochore. And just to illustrate where this is found, here's a chromosome, here's a chromosome. This region here where the two sister chromatids are attached is known as the centromere. Not to be confused with the centrosome, which is very confusing. So don't be beguiled. Okay, and then in the centromere, there's a little sticky region here called the kinetochore. And that is the place, I mean, it's probably not actually sticky, but it sticks to the mitotic spindles. That is the attachment site. All right, that's all happening pro-metaphase. And once the spindles grab onto the kinetochore, they can actually push, or they can pull, retract the chromosomes to get them all nice and lined up, which is what happens when we enter metaphase. Now, metaphase is important because it helps the cell to make sure we have all of the sister chromatids, the duplicated chromosomes lined up here. If there's like one hanging out over here, then after this whole process occurs, it's going to be an unequal division. There's not going to be the same number of chromosomes in each daughter cell, which is going to be catastrophic for those daughter cells. So it's very important that all the chromosomes are lined up. How this cell without brains or ears or eyes is able to extend these super long tubes, grab onto some gnarly little hunk of DNA floating around in the cell somewhere, and then somehow line it up so that they're all perfectly in the middle of the cell, I will never be able to understand. I'm sure somebody knows quite a bit about it, but not me. But it's a thing that happens nonetheless. And then once they're all in the middle, we enter anaphase. And in anaphase, the sister chromatids are pulled apart. Once the sister chromatids arrive to opposite sides of the cell, a new nuclear envelope forms around them. Again, how this is accomplished, I will never be able to understand. How a cell can figure out that a new set of chromosomes has just arrived and now it needs to build two separate little nuclear envelopes. Serious cell signaling going on there to make that all happen. But once those two new envelopes reform, then this is telophase. And also, as it reforms, then the chromosomes can, no longer are they called sister chromatids, they're called individual chromosomes. The chromosomes can decondense and relax after their stressful journey being dragged across the cell. All right. The last thing that needs to happen then is cytokinesis, and in cytokinesis, basically you have the two new nuclei on either side. You have two sets of organelles kind of spread out on either side, and then the last thing that remains is just the physical separation of that cell. This is a scanning electron micrograph that shows what a chromosome looks like. This big little poofy looking thing almost looks like a poodle. And here I just bring this up because you can see over here, this is the centromere. This is the place where the two sister chromatids are bound, and on there would be a kinetochore. Okay, this is just a really cool little figure showing the centrosome. All of these fibers here are staining in green. Those are the mitotic spindles. The blue represents the DNA, and then the red specks are the kinetochores, and it's all these little fibers grabbing onto these little kinetochores. Now, the way a cell undergoes cytokinesis varies depending on if it's an animal or a plant cell. In an animal cell, basically, because it's a little bit more flexible, it just has a plasma membrane. There's a little contractile ring that forms around here and literally just squeezes and cinches it until it boop, pops up into two different cells. This is quite a bit different than in a plant cell. In a plant cell, it's more rigid because of that cell wall. So rather than pinch it until it forms two cells, they literally just build, it's almost like a brick wall. They just build a wall in between, in the middle. It eventually just becomes its own little partition to separate the two cells out. A quick discussion here about prokaryotes. Prokaryotes, an example of these again would be bacteria. Bacteria are interesting because they only have a single chromosome. So they are not diploid. They don't have a mom and a dad that gave them sets of chromosomes. They just have a single chromosome. And because of that, it makes the division much simpler, right? There's none of this business pulling chromosomes to and fro, getting them lined up just right in the metaphase plate, and then pulling them apart in anaphase and reforming nuclear envelopes and all that stuff. So this is a very, very fast process. And I've heard it said that E. coli, Escherichia coli, if they have all the suitable nutrients and environment, and it maintains that way, they can divide once every 20 minutes, which is remarkable. Mitosis in a eukaryotic cell will usually last a day or so, but this can happen 20 minutes in a bacterial cell. I won't go through all the details of this process, but I did want to show you this video just to give you some idea of how fast this can occur. Now this is sped up, but just for reference sake, in 24 hours with this process occurring, one bacterium could form ultimately this number, which is so big I don't even know what it is, but that number of bacteria. So quite a phenomenal feat here in the bacterial species. This process is known as binary fission. And binary fission is not mitosis, nor is it meiosis. It's just something totally different. Similar in some respects, but in large part quite a bit different. All right, we're going to finish our lecture here talking about cancer, because cancer, really what it is, it's a disorder that affects the cell cycle, or rather when the cell cycle is affected, it forms cancer. So to understand how this is the case, consider this. There are 3 different checkpoints, if you will, on the cell cycle. And they're illustrated right here, right here, right here. A cell should not be able to divide if it's not ready to divide, because if a cell divides and it's not ready, those 2 cells are going to be a little goofy and they might cause some weird stuff to happen. So there's these checkpoints here that help to ensure that the cell is ready to divide when it does divide, and it also checks to make sure that the cell, as it's dividing, is dividing properly. So what the big events, the big things that are assessed at these checkpoints: 1, at checkpoint number 1, it's are there enough building materials, basically. Does the cell have enough ATP? Does the cell have enough of its amino acids and carbohydrates and all those things? If so, then it can progress on, and the DNA can be synthesized. And then the organelles can be synthesized. And then in the G2 checkpoint, the big thing that's assessed is the DNA copied correctly? Are there any mutations that occurred? Is every last bit of that DNA accounted for and copied as well as it possibly can? If there's mutations, can the mutations be fixed? If the mutations can't be fixed, it might be the case that cell is basically told to undergo apoptosis, which is an important word to understand. Apoptosis is basically just a programmed cell death. And it's actually to our benefit that cells undergo apoptosis if they're not healthy cells. That's what happens at the G2 checkpoint. At the M checkpoint, there's a big one here. The big thing that's assessed is, are all spindles properly attached to the kinetochore? Again, this would be a pretty big problem. We wouldn't want to complete mitosis if we're at this point here where we've got all of the sister chromatids lined up at the metaphase plate in metaphase, and we've got a spindle here, a spindle here, a spindle here, and a spindle here. And we have a spindle here, and a spindle here, and a spindle here, and a spindle here, and a spindle here. You can imagine that if there was no spindle on this particular sister chromatid, as these start to pull apart, you'd have, this sister chromatid coming this way, you'd have this sister chromatid coming this way, you'd have this sister chromatid coming this way, and vice versa on the other side. But this particular set right here, because this one didn't have a spindle on it, both of those sister chromatids would actually get pulled to this side of the cell. And so this side would, over here, would not have that chromosome, and this side over here would have two sets of that chromosome. Well, that is a very problematic thing. A cell generally can't survive if it doesn't have all of its genes. So it's important that every kinetochore has a spindle attached to it before anaphase occurs. So that's what's going on here at the M checkpoint. And there's a whole bunch of other stuff going on at these checkpoints, but just for our purposes here, those would be the summary. Now, one of the things that will break down a cell if it's not ready, so if one of these checkpoints isn't met and the cell is actually supposed to cease dividing or program itself to die, it will activate a tumor suppressor gene, and a tumor suppressor gene will make a tumor suppressing protein, and the tumor suppressing protein will hopefully shut the cell down. It will likely be a series of proteins and all interacting with one another to shut the cell down. And generally how cancer is formed, how it originates, is that there's something that happens to one of these tumor suppressor genes, so that the tumor suppressing protein that's made from the gene is not working, and so then cells that should have been shut down because they didn't meet all the criteria of the checkpoints, those cells are continuing to divide, and furthermore they are uncontrollably dividing. They don't stop dividing, they just keep dividing. They can't be shut off. That is an example of a tumor, a cancerous tumor. Now, the way that these genes can be altered such that they don't produce proteins that shut down the cell when it needs to are twofold. One way is that somebody could inherit a mutation in their genome. So from the day that they're born, they just don't, there's something a little bit inadequate about the proteins that they produce to shut down the cell cycle. The genes that they have are missing a part, so the protein that they make doesn't quite do the job. An example of this would be the BRCA1 protein, which is something that suppresses the formation of cancerous cells. And Angelina Jolie actually inherited that mutation, and so because of it, she just had a tumor suppressing protein that wasn't doing its job. She was much more prone to cancer. Specifically, this is one that that operates to reduce cancer in specific tissues, namely the ovary and the breast tissue. So she had those parts removed because this particular protein wasn't doing its job, so she was much more prone to acquiring cancer in those areas of her body. Okay, contrast that to then environmentally induced changes. This would be an example here, would be something like UV radiation in sunlight. So the UV radiation, it hits, goes through our cells, it hits DNA inside nuclei, and if it hits a particular gene that codes for a tumor suppressor protein, then that gene might get mutated, and then it no longer makes a protein that does what it's supposed to do, and then the cell cycle continues when it's not supposed to, and it forms a cancerous cell. A pretty common tumor suppressor gene here that's mentioned in this respect would be the P53 gene, which codes for the P53 protein. And just to give you an illustration again of how this works, is that if there is DNA damage and there are cell cycle abnormalities, this particular protein should detect those. And then there are two options. It will either, well, first it will just stop the cell cycle. It will just freeze it. And if DNA can be repaired, it will be. And then if it is repaired, the cell cycle can then restart. If the DNA cannot be repaired, and there's something strange there that can't be fixed, then P53 should tell the cell to undergo apoptosis and destroy itself. But if UV radiation hits one of the genes that makes this protein, and so that the protein is now a little bit misfolded, there's something a little bit strange with it, then if there is DNA damage and cell cycle abnormalities, this does nothing, or it does not adequately arrest the cell cycle, and so the cell cycle continues when it shouldn't. That can lead to the formation of cancerous cells. This is generally what happens in skin cancers. All right, we'll finish up here just talking about various treatments that can be used to treat cancer. One is chemotherapy, and what chemotherapy is basically just an infusion of agents or compounds, I guess you could say, which destroy dividing cells. So it's actually pretty broad. It will just go through and destroy any cell that's undergoing a cell division. The thought being that the cells that are really pretty important, the vital ones in your organs and such, those generally aren't dividing. They're just doing what they're supposed to be doing, like your neurons and things. And the ones that are actively dividing all the time, usually we can afford to kill off a generation of them, and then as you progress through life, those cells should reseed themselves. They should continue dividing. But then also, if cancerous cells are dividing rapidly, it could be the case that when this compound is administered, it destroys all of those cancerous cells, because all of those cancerous cells are cells that are in fact dividing. So this one can be effective, but it also can lead to a lot of collateral damage because it's killing a lot of other cells that are just normally dividing naturally. Another way is to actually go in really and put an instrument really close up to where the tumor is and zap it with some radiation to try and destroy all of those cells. And this can be done more local, so they can basically block off what part of the body receives the radiation, and then hit that tumor, hit the cancerous tumor with the radiation, and hopefully destroy those cells. And a really interesting, more new approach, relatively speaking, is immunotherapy, and this is using the immune system to attack cancer. Your immune system, in the same way that it protects you from invasion of virions or virions and bacteria, it also can recognize abnormal cancer cells and destroy those. But generally what happens is that when your body mounts an immune response to cancer, it's effective for a while, but then your immune response wanes and it gets less strong, and then the cancer cell still survives, and then it replicates again, and then it just overpowers the immune system. So there are a couple of different ways that we can go about making it such that these types of therapies are more effective, and we won't go into all the details here, but that we might save that for a future activity. But in either case, the general premise being that it's your very own immune system that is employed to attack the cancer. That's all I've got for you. We shall continue on next time with a discussion about meiosis.